Welcome to this educational video on chronic kidney disease and the microbiome. With information and perspective, I promise you'll find nowhere else. If you're familiar with my videos, welcome back. If you're new, buckle up. I'm going to do something a little different this time. I'm going to do away with my green and orange charting of the keystone taxa from my meta-analyses, and instead I'm going to focus more on quotes from the authors. That way, you can hear everything in their words. I'm also going to highlight the authors themselves. I want to give you a feel for the breadth of international knowledge of the microbiome, and not have you think I'm interpreting the data incorrectly or misleading anybody. You're going to see from the words of these many authors that they are saying the same things I've been telling you in video after video after video. The same themes are going to pop up, ones with which you are becoming familiar. For example, there are the opportunistic pathogens I'm always talking about, best exemplified by E. coli. I touch on them in every video. These are the bad guys who drive gut inflammation, intestinal permeability, and subsequent systemic inflammation with immune dysregulation. They produce inflammatory compounds like the LPS I'm always highlighting. Then there are the health promoters I constantly mention, best exemplified by the superhero of the gut, F. Prausnitzii. These are the oxygen, pH, and antibiotic-sensitive butyrate producers who truly determine health by multiple actions, butyrate being the most prominent. And here is a slide I've been showing you for many months in my videos. This is my collection of the true health-promoting bacteria in the microbiome. Note, Lactobacillus is not here. I've compiled this information after many thousands of hours of work as the former director of medical education for a microbiome firm and now with my own platform. All of these here are butyrate producers except for bifidobacterium. All of these here except for allostipes and bifidobacterium make butyrate from the fermentation of carbs/sugars locked behind bonds from sources we call prebiotics. These are the true health promoters, and each time a paper in this presentation references taxa, we'll quickly revisit this slide to reinforce this all-important theme. And here are the opportunistic pathogens I'm always discussing. They are consistently bad actors across multiple conditions/diseases. Their prevalence may vary from condition to condition, but in the end, these are the main players in the dysbiotic microbiome. Yes, there are others, but most of the data points fall here when healthy controls are compared to those with a given disease/condition. Let's do a quick review. Within the genus Agrothella, the main culprit is E. lenta. E. lenta is in the phylum Actinobacteria, which is where bifidobacterium is located. Enterobacter has various species with not one in particular acting as the principal bad guy. This LPS-containing genus falls within the very ugly family Enterobacteriaceae. Enterococcus has more than these two species, but these two always come up in the data. These two are particularly troublesome, and this genus falls within the same order as Lactobacillus, called Lactobacillales. Next we have the genus Escherichia, almost always represented by the poster child for opportunistic pathogens, E. coli, which contains a highly inflammatory LPS and is also within the very ugly family Enterobacteriaceae. Fusobacterium is our next genus, which has many problematic species, best represented by the species F. nucleatum. The genus Klebsiella is a genus of a number of species, but pretty much always represented by its very troublesome species, K. pneumoniae. It too contains LPS and lies within the family Enterobacteriaceae, and now you can understand why you never want a high abundance of this family. Your so-called probiotic Lactobacillus, which has had a number of species reclassified to other similar-sounding genera, is next. We'll include all of the traditional Lactobacillus species here, of which there are many. No one species dominates the ugly data. It falls within the order Lactobacillales, along with the two proven opportunistic pathogens, Enterococcus and Streptococcus. Don't you think they could share some pathological similarities? See my video entitled Lactobacillus probiotics, a dumb idea in those who are dysbiotic. Mediterraneibacter is our next genus, which encompasses two troublemakers, especially R. navis, which I mention often. Streptococcus, as in strep throat, is a large genus with many pathogenic species, without one dominating the ugly data points. Again, in the same order as Lactobacillus. Lastly, we have Veillonella, which is an interesting genus, as it is classified within Firmicutes, but contains LPS and is a proven pro-inflammatory opportunistic pathogen. These are the bad guys who dominate the average dysbiotic microbiome. When the environment of the microbiome favors them, they become opportunistic and drive inflammation. We'll also revisit this list with each new set of data points from each paper we will review. We begin with this meta-analysis from these authors published in 2021. A meta-analysis is when you look at multiple, previously published individual original research papers to try to reach a consensus. Here they included 25 studies with a total of 1,436 CKD patients, chronic kidney disease. They found a significantly increased abundance of Escherichia, that's where E. coli comes from, and Streptococcus and CKD, and Streptococcus and Fusobacterium in end-stage renal disease, while a significantly decreased abundance of Faecalibacterium and Roseburia in CKD, and Carpococcus and Faecalibacterium in end-stage renal disease. Here, they highlight how the butyrate these health promoters produce is protective of the kidneys. And here, they mention how the toxic metabolites, p-cresyl sulfate and TMAO, are higher in kidney disease. Sounds like my other videos. So, we take my established list of health promoters from years of work, and we add 3 more data points to further support this perspective. Now, you have to understand, this is data from a meta-analysis, so it carries more weight than from a single paper, as the data was consolidated from 25 individual studies. So we give these stars a dark green color. And likewise, the data points from a meta-analysis for the opportunistic pathogens also carry more weight, so they will get the dark orange color. Here we have another meta-analysis, this one published a year later in 2022 from these authors. They include 15 studies with 1,640 subjects. The comparisons were conducted between diabetic kidney disease, type 2 diabetes, non-diabetic kidney disease, and healthy controls. 8 studies report the differences of gut bacteria. You can see figure C looks a lot like the chart that I use, where green is significantly higher in healthy controls and red is significantly higher in diabetic kidney disease. They use orange to show a difference which was not significant. Here they talk about tryptophan metabolites and how E. coli causes gut permeability and escape of bacteria driving immune response. And as far as taxa, what did this meta-analysis show? Butyrococcus, Faecalibacterium, Roseburia, and Lachnospira were consistently significantly higher in healthy controls, and Escherichia, Enterococcus, and Lactobacillus were consistently significantly higher in diabetic kidney disease. Aratorches was demonstrated to be inversely correlated with estimated GFR of DKD, in other words, a bad thing. We get 4 more data points for the health promoters listed here. Again, this is from a meta-analysis, so if you recall, from figure 3C, there were actually 6 positive data points for F. prausnitzii alone. But instead of listing 6 individual stars, we'll just put the one deep green one for F. prausnitzii and the others here to represent the higher importance of this finding. And when it comes to the opportunistic pathogens, this second meta-analysis gives us 4 more data points for the microbial fingerprint in CKD. The last meta-analysis was published in 2022. So I went looking for other kidney disease/microbiome papers published later, which were not included in either previous meta-analysis. We start with this one from 2022 from these authors. Since heart disease is the #1 cause of death in those with chronic kidney disease, they analyzed the microbiomes of 73 hemodialysis patients and investigated abdominal aortic calcification, AAC. Vascular calcification is an excellent predictor of cardiovascular disease. The patients with high AAC scores, a bad thing, had significantly increased abundances of E. coli, R. navis, and lactobacillus, and significantly decreased abundances of ruminococcus. Summaries of their quotes include, LPS is a key factor in the initiation of progression of atherosclerosis. LPS from E. coli and toxic metabolites from amino acid fermentation may accelerate vascular calcification via inflammation. E. coli had the greatest influence on vascular calcification, and how short-chain fatty acids, as in butyrate, are anti-inflammatory. So we add one more data point for the health promoters listed here. Now, since this data point came from one individual study instead of the sum data from a meta-analysis, we'll give it a lighter green color to note the difference. Still important, but less significant. And the same is true here, with 3 more data points for the opportunistic pathogens, just a lighter shade of orange. Here we have a 2023 original research paper from these authors, who analyzed the microbiomes from CKD stage 1 and 2 patients with and without hyperuricemia and healthy controls. They found Eubacterium ventriosum, Agathobacter, that's erectale, Parabacteroides, and Roseburia were significantly lower in the CKD groups. In other words, significantly higher in the healthy controls. And Fusobacterium was significantly higher in CKD. 3 more data points for the health promoters listed here, and 1 more data point for the opportunistic pathogens. Here we have another 2023 paper from these authors, who assessed the gut microbiome by shotgun metagenomics of 2,438 subjects, 292 with suspected CKD. They examined cross-sectional associations of estimated GFR, urinary albumin-creatinine ratio, and CKD with gut microbiome features. If you take a good look at Figure B, you'll see clear divisions between various taxa identified as worse versus better kidney health. They go on to state, quote, "These findings agree with evidence from other studies that short-chain fatty acids are depleted in stool and serum of CKD patients compared to healthy controls, and that short-chain fatty acids ameliorate kidney injury. Short-chain fatty acids may protect the kidneys via several mechanisms, including reducing inflammation, oxidative stress, and pro-fibrotic factors in kidney cells." So what do they find for taxa? Here, red is a positive correlation and purple a negative. Here on the outer circle is CKD. So you want a purple here, whereas the inner circle represents GFR, where you want a positive correlation, in other words, red. I highlighted the taxa that I mentioned with frequency. We see the opportunistic pathogen E. lenta here with a negative correlation with GFR, a bad thing, implying the inflammation it drives results in impaired kidney function. While here, we see the butyrate-producing, health-promoting species Carpococcus eutactus with a positive correlation with GFR, implying that its protective roles within the gut reduces insults to the kidneys, and the butyrate it produces is anti-inflammatory. If we consolidate these data points, we see beneficial data points for Acinetobacter, Eubacterium, F. prausnitzii, Ruminococcus, Roseburia, E. rectale, E. hallei, Carpococcus, and we see an outlier for Anarostipes, and ugly data points for the classic opportunistic pathogens E. lenta and C. bolte. From this paper, we get 15 more positive data points for the health promoters listed here, with the 1 outlier for Anarostipes, and 1 more data point for the opportunistic pathogens listed here. Just a brief pause here in the presentation. If you could just hit like and subscribe, it would really help this channel out. From 2024, we have this paper from these authors who characterized the microbiome in 94 patients with various pathological patterns of CKD and 54 healthy controls. In the figure, we see a significantly higher abundance in healthy controls, shown in blue, for the health promoters Carpococcus, Gemiger, Oscillospira, and Butyroscoccus, while in red, we see a significantly higher abundance in the classic opportunistic pathogens Klebsiella, Streptococcus, and Enterobacter, with an outlier for Ruminococcus. And here are a couple quotes on the inflammatory nature of LPS and how LPS was the most well-known inducer for fibrosis and renal injury. That's 4 more positive data points for the health promoters listed here, with 1 outlier for Ruminococcus, and 3 more data points for the opportunistic pathogens. From another 2024 paper, these authors conducted a comprehensive analysis of the microbial differences between patients with CKD and healthy controls based on a total of 980 samples from 6 references across 3 nations. They found a significantly higher abundance of the classic opportunistic pathogens R. navis and R. torquis in CKD, with outliers in Ruminococcus and allostypes, while they found a significantly higher abundance in healthy controls for the classic health promoters, Ruminococcus obium, which was reclassified to Blautia a few years ago, Carpococcus eutactis, and F. prausnitzii. They go on to state, quote, F. prausnitzii is highly important for the human gut and health. A reduction in this bacterium can lead to a weakening of intestinal anti-inflammatory and immune regulatory capabilities, earning it the title of a key species in the gut. That's 3 more positive data points for the health promoters listed here with 2 outliers, and 2 more data points for the opportunistic pathogens listed here. For our last fecal microbiome original research study, we have this paper here with these authors. 93 individuals were recruited for the study. 73 patients had stage 3 through 5 CKD, including those receiving renal replacement therapy, the CKD group, and 20 were what they defined as age and sex match controls. What did they find? At the genus level, the butyrate-producing bacteria, Lactospira, Blautia, Carpococcus, Anarostypes, and Roseburia, were significantly more abundant in the control group. And for the patients with CKD stages 3 through 5, they had significantly increased numbers of Parabacteroides, Clostridium, Ruminococcus, and Lactobacillus compared to patients with CKD stages 1 through 2. That's 5 more positive data points for the health promoters, with 1 more outlier for Ruminococcus. Now, Ruminococcus may appear not to have the best data here, but we have to take into consideration that even to this day, years after reclassification, R. navis, one of our bad guys, still gets lumped into the Ruminococcus data. After all, the R in R. navis comes from Ruminococcus, where it was originally classified. I see errors like this on occasion in the papers, like the one we saw earlier, where we saw Blautia obium classified into Ruminococcus. I don't have the raw data for the studies, so I can't say for sure, but experience tells me that probably at least one of these negative data points for Ruminococcus is incorrect. And this paper gives us 1 more data point for the opportunistic pathogens listed here. How's that for consistency? And note Lactobacillus, all negative data points. And for extra credit, I threw in this cool paper looking at the blood microbiome in CKD. Yes, there are bacteria or parts of bacteria floating around in your blood, much more than you would ever think. And don't forget to read this quote from these authors, which reinforces this in every video of mine. Now, when it comes to blood microbiome data, you get a lot of oral, skin, and other bacteria in addition to those from the gut. So it complicates things a little bit. But if we focus on 2 important families, that's the classification above genus, we see 2 of the 3 most important ones in the gut microbiome. Here, we see the family Enterobacteriaceae significantly higher in the blood of CKD subjects. This is the family that I always say is the last family you want to see high levels of. Does it look familiar? It should. More on that in a minute. And here we see this family significantly higher in the blood of healthy controls as compared to CKD. Does this family look familiar? Well, it's been here the whole time. This family houses the amazing butyrate-producing, antibiotic, pH, oxygen-sensitive health promoters, Roseburia, erectile, Carpococcus, Beobium, Lacnospira to include E. elegans, Fusicatenibacter, E. hallei, and Anerostipes. And look at all those data points we established along the way. How is that for consistency in data? The fecal microbiome is clearly represented in the blood microbiome, for the better, as we see here, or for the worse, as we see here. So that family I said is the last family you want to have high levels of. Remember the name, Enterobacteriaceae. Well, within this family lie a number of bad actors, and here are 3 of them. These 3 are clear opportunistic pathogens. All 3 produce LPS, which is recognized by the immune system, TLR4 to be specific, where the alarm is signaled for an invader and inflammation ensues. All 3 of these taxa are not what you want to have an abundance of in your gut. They will drive inflammation within and outside of the gut, causing just about every health concern you can think of. And it's not just taxa from this family Enterobacteriaceae. If we look at R. navis, which is gram-positive, it too is a proven opportunistic pathogen. It doesn't secrete LPS, but R. navis synthesizes and secretes a complex glucoraminin polysaccharide, GPS, which is also recognized by TLR4. See my video on Crohn's disease for more. But from this list, let's just take a look at E. coli, the worst among them. This is the profile across all diseases/conditions for which I've done a meta-analysis, and now we can add the kidney disease information as well. You see orange across the board. That means in just about every study across the planet, E. coli is consistently, significantly increased in those who are ill versus healthy controls. This is no coincidence. If you think this is association without causation, just keep your head buried in the sand. I discuss LPS in about every video. It is but one mechanism whereby E. coli is highly problematic. The LPS of E. coli is highly immunostimulatory, 100 to 1,000 times more so than LPS from some other taxa. To reinforce this, let's take a look at an LPS study in kidney disease. These researchers looked at all LPS in the serum of 477 subjects who were followed for 6 years. When looking at progression of kidney disease, those with the higher level of serum LPS fared significantly worse. This implies that the pro-inflammatory effects of a dysbiotic microbiome driving inflammation and gut permeability with subsequent whole or parts of bacteria like LPS entering circulation and driving systemic inflammation and immune dysregulation throughout the body, and here specifically in the kidneys. They also interestingly note the importance of HDL in this equation, as it plays a key role in detoxifying LPS, which I cover in depth in my video on cardiovascular disease and the microbiome. On the flip side, if we look to the superhero of the microbiome, F. Prausnitzii, we see virtually all green, signifying that when a significant difference was found between those subjects with a disease/condition versus healthy controls, the healthy controls consistently had significantly higher abundances. Again, this is no coincidence. I have a whole video dedicated to this amazing species, which dives into the whys and wherefores. And as F. Prausnitzii in the healthy is about 5% of the microbiome, it's on average the main butyrate producer in the gut. And as butyrate is such a critical molecule to health, let's take a look at an in vitro study. These authors took endothelial cells from the kidney and cultured them with both butyrate and acetate, 2 short-chain fatty acids. In figures F and G, you can see how butyrate, but not acetate, significantly increased claudin expression. Within their paper, they have many statements linking LPS, nitric oxide, and butyrate, topics I discuss in my videos. As I say, it's all connected. The value of HDL and nitric oxide not only apply to kidney health, but they apply equally to cardiovascular health. See that video. They also mention permeability of the blood-brain barrier and its connection to LPS and butyrate, which ties in with anxiety, depression, clarity of thought, Parkinson's, and dementia. All topics I cover in my videos. So now perhaps you're beginning to understand that the principles that apply in one part of the body also apply in another. I don't view each disease as its own separate entity as much as I view them all as oxidation and inflammation. So let's take this universal connection one step further to wrap up this video. This paper before us highlights the claudin protein and its role in endothelial cell permeability. But where have we heard that claudin word before? If you saw my video on gut permeability, you may remember the figure to the left. That's a diagram of the enterocytes in the gut, a model of a healthy gut and one with permeability. Look closely. You see claudins as one of the many proteins keeping the integrity of the gut intact. To the right, we have a figure from a paper entitled Claudins in Renal Physiology and Disease. We see claudins here responsible for epithelial cell barrier integrity. We also see the other proteins, occludin, jam, and actin, which are also in the image to the left. Permeability is not only relevant to the gut, but many other parts of the body. The job of these proteins is to be selectively permeable for a variety of tissues, but when damaged, this control is compromised. The need to maintain this barrier integrity exists in many parts of the body to include, but not limited to, the gut, liver, skin, lungs, kidney, and brain. You've probably already heard of the blood-brain barrier. Well, that's one of many. And from this paper, we get the following quote. Quote, "At the blood-brain barrier, claudin 5 is the most enriched tight junction protein, and its dysfunction has been implicated in neurodegenerative disorders, such as Alzheimer's disease, neuroinflammatory disorders, such as multiple sclerosis, as well as psychiatric disorders, including depression and schizophrenia. Therefore, in order to support systemic permeability, we should first look to the number one source of microbial-driven inflammation, the gut." In this video, I did not dive into ammonia, p-cresol, and indoxyl sulfate, all very valid subtopics here. I do cover them in other videos. It's nearly impossible to touch on everything and still keep these videos a reasonable length. But when you watch enough of them, the light bulb will go off, and you'll realize that your health starts in the gut. It is no coincidence that antibiotics and PPIs will kill off the health promoters, and over time, the opportunistic pathogens will be in charge. I hear all the time of how antibiotics are the trigger to someone's downfall. Dysbiosis will drive inflammation within and outside of the gut. The leakage of various compounds drives systemic inflammation and, on a chronic level, immune dysregulation. If you're concerned about kidney health or any other aspect of your health, you need to address the root cause, the microbiome, with an intelligent approach, not the same old ridiculous probiotics and fermented foods. Ask yourself, where else have you seen this information before? I'll bet nowhere. I know this material like nobody else. I help legions of people through a background and perspective that's different from anything else out there. Watch my videos. You'll see the same themes repeat themselves, just like the consistency you saw in my data points from earlier. This consistency exists in the published literature. It's just the science. It's the biochemistry of us. If what you've been doing hasn't been working, reach out and I'm here to help. Well, I hope you enjoyed that video. And I will say that basically every single day on my YouTube comments and in emails, I get a lot of thanks from people out there who really appreciate the information I've been sharing. And so part of that is you're welcome. The other part is you can contribute by clicking on the super thanks below. And if you're not doing a consultation with me and you're not purchasing any protocols, it's a great way to support this channel. Each presentation, depending on the presentation, but most of them take an incredible amount of time to put together. There's a lot of material. There's a lot of data checking. And so it's just sometimes 50, 60, 70 hours to put together one presentation. And so if you can just click that super thanks, I'd appreciate that, and we'll keep the information coming.